Welcome back, everyone, to Kathy Nagel's second talk. Over to you, Kathy.
Well, thanks, Sanjay. Welcome, everybody, to today's second lecture. So let me share my screen. Can everybody see one slide? Does it look okay, Sanjay?
Yeah, we're good.
Okay, great. So thanks everybody for coming. So today I'll be talking about some work mostly from our lab, trying to understand how flies navigate in odor plumes. And it's wonderful to be giving this talk in this course with, since I think previous talks have introduced all the pieces needed for this talk. So I think Antonio Solani talked about olfactory odor plumes and plume navigation. Dima introduced the olfactory system, and Larry introduced some of the navigation circuitry in the fly. So I hope to just sort of pick up on what they've introduced and talk about how we've been studying this in a particular context of a particular navigation behavior in the fly. So just to reiterate what other lecturers have introduced, using odors to find things in your environment is sort of an incredibly ancient and conserved behavior. So this is found even in organisms without brains. So bacteria will use odor cues to move up odor gradients. Insects are excellent olfactory navigators. Fruit flies, which we study, ants, mosquitoes all use odor cues to find food sources in their environment. Obviously mammals use odor cues to find food sources as well. So the wolf shown here, or dogs, very obviously are using their noses, using the sense of smell to find things in their environment. And part of the reason that we like this problem of olfactory navigation is that unlike something like visually based navigation, where the location relative to the animal of a target is kind of obvious at the first sensors, olfactory navigation always involves some kind of internal processing. So typically as an animal moves through an odor environment, what it's experiencing is changes in the concentration of odor over time. And it has to translate those concentration changes into a pattern of movement or a sequence of locomotor actions that will take it to the source. So this has been, famously studied in bacterial chemotaxis. Bacteria mostly navigate in odor gradients. The general model is that they care about the dynamics of the odor signal. So when the odor is increasing or steady, they keep going forward. And when the odor concentration drops over time, that elicits an increase in turning probability, which sort of gives them a biased random walk, which leads them to the peak of the gradient. And I think Antonio introduced the idea that for many macroscopic organisms, olfactory search is really different, and that's because of the physics of the odor environment. So when we encounter odors, or a dog, or even a fly, we're not encountering a smooth gradient like I showed you in the last slide, but we're encountering a turbulent plume. And this is an image of a plume taken by our collaborators in the Grimaldi lab at the University of Colorado Boulder. This is actually an acetone plume being imaged with a UV laser light sheet. This is at about the level close to the ground where a fly would be walking. And this is sped up a few times. Can you see that moving, I hope?
Not really. Yeah.
No. It may be a little bit delayed.
Yeah, it's moving. Yeah, it's moving.
Okay, so what I hope you can appreciate about this, so this is a plume. There's a wind source from the top, and what I hope you saw in the video, at least briefly, is that this is always in motion. And so what a fly would experience as it's walking through this is not a smooth gradient, but many fluctuations in odor concentration. And this means that the concentration itself, or even the derivative of odor concentration, is not necessarily a good cue about which way to go to find the source. So this problem has been studied for a long time. And the way that organisms solve this problem is actually quite elegant, which is they don't depend just on the odor concentration. They also pay attention to the wind direction. So this was first worked out in moths in the 80s. This is a really beautiful experiment where a moth was released. Moths will track pheromone plumes. Male moths will track pheromone plumes over miles. So they released a male moth at one end of a field, and at the other end of the field, they released a female pheromone next to a bubble maker. And so they could use the bubbles to track where the plume was. And what they observed was that when the moth was in the bubbles, it would tend to orient upwind. And when the moth was outside the bubbles, it would tend to orient across the wind. And this led to the idea of odor-gated anemotaxis, that is odor-gated wind orientation, where it's really the wind direction that's telling the animal which way the source is, and odor is telling you whether to orient upwind or whether to orient with some other orientation to the wind. And so what this really means is that in order for a fly to navigate towards an odor, they must be able to flexibly combine two different sensory inputs: an olfactory input that tells them whether to approach something or not, and a wind direction input that tells them which way to turn. And so when I started my lab, which is now like 7 years ago, I think, we thought this was a really beautiful behavior to focus on and to take advantage of the fly circuit cracking toolbox, which I talked about in my last lecture, to really work out neural circuits that might be underlying this behavior. So that's what I want to talk to you about today. I'll have sort of 3 parts to my talk. I will say up front that we haven't figured out the entire circuit yet, but I think we've made a bunch of progress in understanding how neural circuits that perform this behavior are organized, and we've learned some really interesting things along the way, as well as opening up new questions that we hope to address in the future. So in the first part of my talk, I will talk about a behavioral apparatus that we developed in order to study olfactory navigation in fruit flies, and how that led us to develop a sort of algorithmic model of navigation. Next, I'll talk about how flies sense wind direction and how those signals are computed and transformed and represented in the central brain. And finally, I'll talk about olfactory circuits in the fly and how those are organized and where they impinge on the wind direction circuit. And if you have any questions, please put them in the chat, and I'd be happy to address them as I'm going. All right, so the first part of my talk is about measuring and modeling olfactory navigation behavior. And this is mostly the work of a former postdoc in the lab, Efrain Alvarez-Salvado. It was published a couple of years ago. So first, I just want to show you that flies can actually do this task that I showed you. This is a turbulent wind tunnel. The difference between is that, so this is a large wind tunnel. It's about a meter long and a third of 30 centimeters wide and tall. And because it's sort of open to the air and the grid in the front of it, it's got turbulence in it. We're looking only at walking flies at the moment. The question of how they navigate in flight versus walking will come up a little bit later, and I'm happy to address that, but we just glued their wings down so that they couldn't fly. And we basically, so the movie that I showed you a couple slides ago was from our collaborators in Colorado, and we built the same wind tunnel here in New York so that we could essentially study behavior in the same, at least statistically, the same type of environment. I know the videos aren't working super well. I'll try to play this here. Oops, maybe it's not going to work. Sometimes it works better if I come out of share mode. So I'll try playing this here. Okay, can anybody see that moving? So this is a large wind tunnel with a tiny fly in it. So there's a tiny fly that's a spot here that I hope you can see. And Efrain has overlaid here the area that our collaborators were able to image with the plume image. So of course this thing is moving, but this is just showing you where it is. And what I hope you can see is that the fly does make her way up the plume and is able to find the source, but that this isn't like a trivial behavior for the fly. She doesn't run straight up. It's actually challenging behavior for her. And let me see if I can play that again.
It's hard to see the fly.
Yeah, she's very tiny. So this is it gives you an idea of the scale of this thing. So there's a tiny dot that's moving around here. But you can see her sort of searching around. She'll make a lot of different turns and sort of stop and go as she's making her way. But she does sort of stay within the confines of the plume and make her way there. So this is just to show you that one, flies can do this task, but it's challenging for them. And we think that's actually an advantage experimentally to give them something that's challenging. But of course, one of the problems with this setup is we don't actually know exactly where the odor is at any point in time or what the fly is experiencing moment by moment. And so here, let me go back to
The slides for some reason look very pixelated. I don't know if others are having this.
Okay. Okay. Do you see now? The video should be gone and there should just be a diagram. Do you see that?
Yeah, but yeah, now it's better. Yeah, it's better.
Okay. Yeah. There's a question in the chat. Was any noxious odor also provided while this was being done to see if the fly avoids it? No, so this was just a plume of attractive odor. We haven't done a lot to date with aversive odors, although I'll show you a little bit later that we have some sort of aversive neurons within the brain. And it's something we're just beginning to explore now. Actually, a new graduate student in the lab is starting to look at a variety of different odors. It's a great question. Okay, so to get around this problem of not knowing exactly what the fly was experiencing, we developed this miniature wind tunnel system, which, unlike the previous system, allows us to precisely control the odor and wind stimuli that the fly experiences as it's walking around. So this is basically like a little sandwich of plastic. The fly can only walk within it. And the other thing is that the length scale here is small. It's only, a 1.5 millimeters tall enough for a fly to walk around. But that also means that the airflow within this this miniature wind tunnel is constrained to be laminar. And that means that we have the ability to, exactly predict the movement of the air and when the odor comes on. And so we have a constant flow of air, so a constant wind direction from the top here. And then we can inject into that essentially a front of odor that's going to move through the wind tunnel. And so we know exactly when the fly hits the odor and when the odor stops relative to the fly. And so we can essentially do sort of open loop sensory physiology with behavior in the fly, and then we can track their behavior as they're walking around. So this is just to show you that we have excellent stimulus control here. The wind speed is constant throughout the experiment, and then the odor turns on and off with very precise kinetics. I have another video here, which again may or may not work. I'll just have this play in the background. But what we observe is that flies like to hang out at the downwind end. They prefer to go downwind when there's no odor. And then when the odor comes on, which is shown by this green light here, they actually run upwind. And when the odor stops, they'll start to do sort of a search behavior around the place where they were when the odor stopped. That can be seen a little bit more easily here in these trajectories. So this is again just showing you what I just had a video. So in black here are the trajectories of the flies in the presence of wind but the absence of odor. And they like to, they prefer to occupy the downwind. So they have a sort of a mild downwind preference in the absence of attractive odor. Then when the odor turns on, they'll turn and they'll run very rapidly upwind. And after odor offset, they'll start to do this sort of search behavior around the odor off, the location where they were when they lost the odor. And we can get a large number of trials very quickly from this apparatus. And so we can make measurements of sort of average fly behavior as a function of time. So up here, I'm showing you again my stimuli with constant wind and with a pulse of odor with nice kinetics. And you can see that during the odor, the upwind velocity increases sharply. There's some adaptation and then they go downwind after the odor stops. And we can also measure the curvature of their trajectories. That's just the angular velocity, the absolute value of the angular velocity over the forward velocity. So a measure of sort of tortuosity or how curved the paths are. You can see that there's a peak at odor onset, and that's because many of them turn to face upwind. They actually straighten out during the odor. And then after odor offset, there's this large increase in curvature, and that corresponds to this search behavior that you see here. Okay, so we next wondered whether these two behaviors together could account for how flies find a real odor source in a turbulent environment. And so we developed a fairly simple computational model of this behavior, which I'm going to illustrate here. As I go through this model, one thing is that we knew a lot at the time that we started this about sort of the front end of the olfactory system, work that I did and that many others have done on olfactory receptor neurons and how they encode odors, taught us a lot about sort of encoding at the front end, at the periphery. And by contrast, at least at this time, we didn't know very much about the motor end of how they actually generate motor commands. So the motor end of this is going to be a little bit sketchier and is probably inaccurate. And we're working on updated models now. But we think that the front end of this is actually not a bad match to what the flies actually do. Okay, so the model is that you have some fluctuating odor stimulus that's coming into the system that could be because we're presenting it as an experimenter or because the fly is moving itself through its environment. And based on what we know happens in olfactory receptor neurons at the periphery, we imagine that there is a transformation of olfactory information that happens at the front end. We call this adaptive compression. It's compression because it goes through this sort of a Hill function, which comes out of the binding of odor to receptor, which gives us a response that's sort of a sigmoid as a function of log odor concentration, and that lets the fly deal with the huge range of odor concentrations that it encounters as it's walking around. And then there's some adaptation in the system, and again, this has been well documented, and it actually arises from many stages of processing in the initial sort of neurons and synapses of the system. But the basic idea here is that in the presence of sort of continued odor, the sensitivity of the system is reduced. That is, the odor response curve gets shifted to the right, meaning that it takes more odor to get the same response from the neurons. And what this means in practice is that over time, the response will adapt or decrease a little bit, and it will reset their sensitivity so that essentially they'll never get saturated. And in this model, we set this to always come to sort of the mean of the local average of the odor fluctuations. Okay. So odor gets sort of adaptively compressed at the front end, and then it goes through two temporal filters, one of which we call the on filter and one the off filter. So the on filter is going to turn on whenever the fly is embedded in the odor, and importantly, it's got some width to it, so it's going to smooth out some of these fluctuations. So it's basically going to say, yeah, I think I'm inside the plume now, even if the plume itself is going up and down. And the same is true of the off filter. This is sort of a differentiating filter, but importantly, again, it has sort of a long integration history. And again, what that means is if the fly is embedded in the plume and it's going up and down, it's not going to treat every one of those fluctuations as, oh my goodness, I lost the plume. It's only when it's been in that and then it really loses it for a long period of time that it will initiate something like search behavior.
There's a question.
Okay, question. In the last slide, is the before odor time interval in the third box significantly lesser than the other boxes? There doesn't seem to be a lot of movement in that phase. It's also interesting that after odor trajectory, it's almost exactly the opposite of the odor phase, unlike the seemingly random movement in the first two boxes. Is there a reason for this? Let me just go back. Oops. Okay, so there's a bunch of questions in here. Right, so I didn't show it here, but actually, in addition to generating this upwind movement or upwind orientation and sort of change in velocity, the encounter with odor actually makes them move more. So if we look at just like the probability that they're moving at all, that goes up in the presence of odor and actually lasts for a long time after odor. And this a very astute observation here. They're actually moving less in the black phase, in the pre-odor phase. The after odor trajectory is so the after odor trajectories are really variable. They're incredibly variable across flies, across like different genotypes and things like that. And you have to take this downwind thing here with a little bit of a grain of salt. Part of that is because they've hit the top end of this thing sometimes, and they do have this sort of baseline downwind drive that we've measured. So we think that this isn't necessarily kind of a reaction to odor offset. This is just sort of the baseline downwind drive taking over. And this whole phase sort of is slower and lasts longer than the odor phase. But there's some subtlety in that part. And we're still sort of working on exactly what's going on in that phase. Great. Okay, so coming back to this model, the odor goes through this adaptive compression. It gets filtered into two internal signals on and off. And again, we've encountered neurons in the fly brain that do this, that either respond during the odor or respond basically to odor offset. And then we imagined, because at the time that we built this, we had no data for this, that each of these sort of internal signals on and off could drive different locomotor components. So on would drive upwind orientation, it would drive an increase in forward velocity, and it would suppress turning, leading to straighter trajectory. Okay, so I walked through the model. Okay, so we wanted to test this model. And did you lose the end of the model explanation, or was I still on for that?
Can't remember. I think we got that. We can move to the next slide, I think.
Okay, great. Yeah. So we wanted to test whether this model could account for the behavior that we saw. And so we did a couple different tests. We first asked whether the model could recapitulate the behavior that we observed in our miniature wind tunnels, where we gave just a pulse of odor. And so these are now model flies responding to a pulse of odor. And you can see that just like the real flies, they tend to go downwind in the absence of odor. During the odor, they run upwind, and afterwards they do sort of a local search. So that was great. And we did some further tests to show that, for example, we could actually account for some of the variability that we observed across flies using just subtle changes in this model. But the real question is, can this model, which was derived from behavior in a very constrained environment, predict behavior in a more natural environment? And so to do that, we turned to these plume movies that our collaborators had collected in Boulder. And as I said, we built the same wind tunnel essentially here in New York, relying on the fact that if you build essentially the same wind tunnel, you get the same statistical plume. And we did some measurements with a photoionization detector to verify that this was true. So this is just the plume again. So this is another video. We'll see if this works. But here I have a spot that represents the model fly. And now it's running in, not in a pulse of odor, but in a plume. And what you'll see is that when the dot is pink, the on function is dominating and the fly is making its way upwind. And when it turns blue, it is the off function that is dominating, and the model is doing some kind of a search behavior.
Kathy, the slide is looking a little bit pixelated. I think there might be a network issue. Maybe you could try cutting your video.
Okay, so we'll just skip the video.
No, your the speaker video. If right now it looks fine, but periodically it goes into this. Yeah, that's better. Let's see if this works.
Okay.
Okay.
Let me. Oh, I just lost you guys. Can you still see me?
We can see you. Oh,
okay.
I mean, we can see the slides.
Okay. Yeah, I think it's struggling with videos. Okay. So let me. Okay, great. Okay, so if you look at the center panel here, this shows you a trajectory of what I just tried to show in the video. So this is an example trajectory of a model fly. So actually on the left here is a trajectory of a real fly in the wind tunnel, and you can see the way that the fly sort of gradually made her way upwind, and then sort of when she encounters the edge of the plume, she'll turn around and make her way back in. And when she reaches the source, she'll sort of dance around the source. And we observe very similar behavior in our model flies, where in a subset of flies, they'll sort of make their way upwind. If they lose the sort of nominal edge of the plume, this will induce the off function and cause them to re-enter the plume. And when they make it to the source, they'll sort of dance around here. And in fact, quantitatively, the success of the model was very similar to the success of the flies. So as I said, this is a hard task for flies, and only about 60% of the flies actually make it to the source. Others sort of get lost somewhere in the middle, or they just go downwind and they never find the plume. And we found very similar behavior with our model. So in the first version of our model, actually, the model did slightly better than the flies. It was closer to 70% of the flies that found, the model flies that found the source. And then we ran another version of the model in which we incorporated sort of cross-fly variability in sort of search ability, search propensity, and that did a little bit worse than our flies. And so we sort of bracket, the behavior of the real flies. And so overall, we found this to be a relatively good sort of first pass fit to what we think the flies are doing. So just to summarize this first part of my talk, I showed you that we built a sort of a high throughput open loop paradigm that lets us measure two basic components of olfactory navigation. One part that we call the on response, which is an increase in upwind orientation and increased ground speed. And the other part, which we call the off response, which is an increase in curvature that causes them to search around where they lost the odor. And I showed you that a sort of simple algorithmic model based on these two components achieves performance similar to real flies. And so we took this as sort of a reasonable justification to now say, okay, let's use this this apparatus and this idea of this on and off function to go into the brain and try to figure out where each of these components might be instantiated. So any questions on this first part of the talk? I apologize for all the video glitching and stuff like that. I can take any questions now. No questions? Okay, so let me go on to the next part of my talk. So we next, when we started to try to work out neural circuits that might be performing this behavior, one of our first questions was, well, how do the flies actually sense the wind direction? And very little was known about this when we started, but I would say actually we now have a pretty good idea of how wind is encoded and transformed in the brain. And so the work I'll be talking about today is really from two people in my lab, Marie Suver, who is a postdoc in my lab, who's leaving, I believe, tomorrow to start her own lab at Vanderbilt University, and Tim Courier, who was a graduate student in the lab, who's now a postdoc at Stanford. So the question is, how do flies determine which way the wind is going? And one thing that I'll come back to is that the way that you sense wind depends on what you're doing. So if you're a walking fly and you're planted on the ground, then wind is largely a mechanosensory cue. And I'll show you in a minute that they mostly sense wind using their antennae. So there's a question from the previous part. Can you derive the true on and off responses from behavior instead of modeling them as arbitrary functions? Yes, so I didn't go into this in detail, it's in the paper, but we actually fit the on and off filters to behavioral data. So we did a series of experiments where instead of just giving a pulse of odor, we gave, for instance, fluctuating odor stimuli or ramps or different things like that. And using and so you can see that the behavior will follow these fluctuations up to certain frequency cutoffs. And we've actually done this up to higher frequencies now using optogenetics. And so we were able to use those to at least fit the on and off functions to the behavior. That's a great question. Okay, so right, so a walking fly is able to sense the wind direction using mechanosensory cues derived from the antenna. But if a fly is in flight, then if the wind blows on it, that will only transiently activate its mechanoreceptors, but then it will cause the fly itself to move. And actually, the computation of wind direction in flight is much more complex because the fly will start to move in a direction that's sort of a vector sum of its own self-propelled motion plus the motion that's being introduced by the wind. And I'll get to at the end of this section a little bit of a thought about how that might be extracted. But anyway, to keep things simple, we decided to start with walking flies, okay? So for the walking fly, they sense wind with their antennae. And this had been known a little bit from studies in cockroaches back in the 70s. But I'll give you a demonstration of this here for Drosophila. So the Drosophila antenna has three parts. This is a zoomed in view of the antenna. There's three segments, 1, 2, 3. And the third segment can rotate with respect to the second segment. And it also has this large bristle coming off of it called the arista, which acts like a sail. So it amplifies small air currents and allows the fly to pick it up. And then there are stretch receptor neurons here in the second segment that can detect this motion and send those signals to the brain. So we can make the fly wind blind by clipping off this bristle and then putting a very tiny drop of glue between the second and third segments, which stabilizes the antenna and makes them unable to sense these wind currents. So we then ask, what happens to flies' behavior if you stabilize the antenna? And that's shown here with some trajectories. So again, here, this is in the wind tunnel, and we're presenting a pulse of odor. And you can see that they can still respond to the odor, which is important because it shows that they can still smell. They actually smell with the third segment of their antenna as well, as I talked about in my previous lecture. So now in response to the odor, they again sort of start moving more and they'll take off in a direction. And they still show this sort of search behavior at odor offset. However, you'll notice if you look at these trajectories that they no longer consistently orient upwind. So here in some examples, they run upwind, in some examples, they run downwind, and in some examples, they run across the wind. And when we quantify this, what we can see is that this increase in upwind velocity is completely abolished when we stabilize the antennae, indicating that they've lost the orientation cue. However, this increase in curvature at odor offset is intact, showing that they can still smell. Okay, so this demonstrates that the entire wind direction signal is coming through that antennal joint, and there's no other wind signal that's getting into the fly. Okay, so how do the antennae actually encode wind direction? So to look at this, Marie Suver in the lab developed a preparation where she could mount a fly at the center of 5 different wind streams, and she had a camera placed below this so she could look up at the face of the fly, and then she could also make electrophysiological recordings from single neurons in the fly brain. So this is a picture of the face of the fly. Here you can see the antennae and the aristae coming off here. This is another video, so this may or may not work. I'll just let this play. And what you can see is that when the wind blows, which is when that green light comes on, the antennae are deflected slightly. Okay. And so this was, again, several years ago. So we quantified this just by having a couple of high school students come to the lab and track the antennae. We're now doing this with deep lab cut, which is much better and much faster. But what we observed is that each antenna is maximally deflected back towards the head when wind hits it from about 45 degrees contralateral to that antenna. And that's because that's the angle at which the wind direction is normal to the plane of the arista. Okay. And then as the wind direction deviates from that, the antennal deflection decreases as sort of a cosine of that angle. And that makes sense just from the sort of physics of the wind impinging on this arista. And so what that means in practice is that each antenna has a displacement tuning curve that's actually gives an ambiguous signal about which way the wind is blowing. So that is, there are multiple directions of wind that give you the same amount of deflection of one antenna. However, you can see that the 2 curves are offset from each other because of the angles of the aristae. And what this means is that if you could take a difference between these 2 antennal deflections, you would get an unambiguous, almost linear code for wind direction. Okay, so Marie set out to find whether there were neurons in the fly brain that could perform this computation. And she began by recording from a set of second-order neurons that we knew from anatomy were downstream of these first-order stretch receptor neurons that we knew could encode sort of tonic deflections of the antenna that we produce during wind. So these are a group of neurons called APNs, specifically APN2. And when she made whole cell recordings from these neurons and presented wind from different directions, what she observed is that these neurons were hyperpolarized by wind, but the amount of hyperpolarization depended on the angle of the wind. And if she plots the steady state tuning curve of the neuron, she got sort of this hook-shaped tuning curve, which very much resembled the tuning curve of the ipsilateral antenna displacement. So that's shown here. This is the displacement tuning of the ipsilateral antenna, and this is the neural tuning of this APN2 neuron. You can see they look quite similar. And so she hypothesized that these neurons were just encoding ipsilateral antenna displacement. And to test that, she removed the ipsilateral antenna, and these neurons go completely silent, which is exactly what we expect. So these second order neurons are encoding just ipsilateral antenna displacement.
So in flies, I guess in flies, like in other insects, the Johnson's organs are projecting in the ipsilateral AMMC, right?
That's right.
Okay.
That's right. Yeah.
And do they all terminate in the AMMC, or do they go beyond the AMMC? Do some of them go beyond the AMMC?
Yeah, so that's a great question. So we did like most of them go to the AMMC. We did some like trans tango on the wind sensitive JONs, and there are neurons that go a bunch of places. We haven't done, we kind of, we reached that point in the project when we decided we needed the connectome to go further. So I'll get, when I get to I think this project is sort of ripe to revisit with connectomics to really understand who's downstream. I think the circuitry is actually quite complex.
Okay.
That's a great question. But it seems like at the level of the AMMC, both from our data and, for example, Rachel Wilson's lab has done some just whole brain imaging, that the signals at the level of the AMMC are really unilateral.
Yeah.
Okay. So then she went looking for neurons that might be integrating signals across the two antennae. And she did this through a slightly crazy, but ultimately successful whole cell electrophysiology screen. So we looked for neurons that got input in this area called the wedge that these APNs project to. But she identified this group of neurons, which she called WPNs, for wedge projection neurons, that have this beautiful linear encoding of wind direction. So they are excited by wind coming from the ipsilateral side and inhibited by wind coming from the contralateral side. And again, if she plots the steady state tuning curve, you can see it has this sort of linear encoding of wind direction, which looks very much like the difference between the two antennae that I showed you earlier. And so Marie hypothesized that these neurons were integrating signals across the two antennae to generate this code for wind direction. And so she tested this by stabilizing either the ipsilateral or the contralateral antenna. And what she saw was that in both cases, stabilizing one antenna reduced the wind direction tuning in this neuron, but did not abolish it, which is consistent with the idea that this neuron is getting both ipsilateral and contralateral input about wind direction and integrating these two signals to generate this linear wind encoding. And actually, if you look closely at the sort of unilateral input to this neuron, it has that same hook shape where there's one that's very different from all of the others. That you would expect from the ipsilateral tuning, and by putting these two together, you're sort of getting this full range of wind tuning. There does seem to be some amplification that's happening in this circuit, so the intact response of the neuron is more than sort of the linear sum of these two. We don't quite understand how that works yet. And so this study was also published a couple of years ago, and so I didn't want to go into all the details of the experiments she did, but she did a further set of experiments to identify the source of, or at least one source of these signals. And she identified a population known as B1 neurons that she thinks is providing the contralateral input to the WPN, as well as these APN2 neurons that I showed you earlier, which she thinks are providing ipsilateral input. And broadly, her data are consistent with the idea that the WPNs are integrating signals from these two populations with opposite sign, but also that there are probably many more neurons that are contributing to the wind direction signal that we observe in the WPNs. And as I said, I think this would be a really nice thing to now revisit using connectomics to really work out all of the inputs to the WPNs. That was sort of our study of how wind direction is extracted at the periphery and computed from a comparison of the displacements of the two antennae. And like I said, this has been a really satisfying field to work in, because when we started this project just a few years ago, we really had no idea where wind was encoded beyond the periphery. And Marie's study, together with another study I'll talk about in a second from our lab, as well as one from Rachel Wilson's lab, have now worked out, I think, at least sort of a skeleton of a complete pathway from the from the periphery to the central brain that encodes and that extracts wind direction. I'd like to just sort of highlight a couple of findings from those studies in the second part of this section. So I believe Larry in his talk earlier today introduced the fan-shaped body, which is a part of the central complex. So it's sort of the navigation center of the fly, and it's quite literally in the center of the fly brain. And Tim Currier, a graduate student in my lab, set out to understand whether wind direction might be encoded in the fan-shaped body. So he set up a recording apparatus where he could, again, record from single neurons in the fly brain while presenting directional stimuli of various modalities to the fly, including airflow, a visual stripe or landmark, as well as odor. And importantly, these were all stimuli that we knew caused sort of orientation responses in the fly. And he then did a survey of inputs to this region called the fan-shaped body, particularly focusing on columnar inputs. These are neurons called PFNs that I think Larry talked about in his talk, in part because we had good genetic tools for targeting those neurons. And what Tim observed is that a subset of these columnar inputs, these PFNs, showed very strong directional tuning for airflow direction or wind direction. That's shown here for one of these cell types. I think these are called PF1 and 3s. So these neurons are strongly excited by wind coming from about 45 degrees ipsilateral, and they're inhibited by wind coming from about 135 degrees contralateral. And as I said, Tim surveyed a large number of columnar neurons of the central complex, and he actually saw wind direction signals in a lot of these cells, but the strongest ones were in a group of PFNs, so columnar inputs to the fan-shaped body, and particularly those targeting the ventralmost layers of the fan-shaped body. So these are distinct from the ones that I think Larry talked about in his talk, which target the more dorsal layers of the fan-shaped body. And so Tim then looked at whether these wind direction inputs could be modulated by odor or the other stimuli, and it looked like they seem to be encoding a fairly pure wind direction signal. So here he's showing the tuning of this group of ventral PFNs to just airflow, and this is in an odorized airflow. And you can see that these tuning curves don't look substantially different, suggesting that they're not modulated by odor. And at this stage, even this is probably 5 synapses into the brain, you're encoding a fairly pure wind direction signal. Now, another question that he asked was, because we thought that these columnar neurons might be representing something about space, and because other studies, I think Larry talked in his talk on Friday about the compass system, we wondered whether different PFNs might be encoding different preferred wind directions. But that doesn't seem to be the case. So Tim made recordings from different PFNs that were targeting different columns of the fan-shaped body. And what he saw was that all of the PFNs on one side of the brain all shared identical wind direction tuning. So all of them were maximally excited by wind from 45 degrees ipsilateral and inhibited by wind from minus 135 degrees. So this was sort of confusing to us at the time, but especially when these studies that Larry mentioned in his talk came out, talking about how PFNs might encode things like optic flow as basis vectors, this sort of made sense to us. So we think that these ventral PFNs are encoding airflow direction as a set of basis vectors. So the one thing that I didn't show you here is that if Tim recorded on the opposite side of the brain, the neurons had the opposite tuning. So those neurons prefer wind from 45 degrees on that side of the brain and are inhibited by the other side.
Kathy, have you or anyone else measured the resolution of this system? How good is it at how much can you change the direction and the fly notices it?
A little bit. So we have these tuning curves, and one could derive from these tuning curves plus the noise, which which is shown as error bars here, the resolution. I will say that we were able to troubleshoot a 1 degree offset in our stimulus just by looking at these PFN responses. So when we first set this up, on subsequent trials, the wind was offset by a degree, and we could see in the responses that it was shifting over. So this was really surprising to us. We typically think of these neurons in the central brain as being, they only respond during behavior, and they're not. But these are very well behaved sensory neurons, and I think they have actually very high acuity. We haven't done like a quantification of that, but that would be my guess. Yeah, they're amazing. Yeah. They're like little machines. Yeah. So and we've also Larry talked about how the dorsal PFNs that encode optic flow are getting both sort of a direction signal as well as a heading signal. And Christina May, a postdoc in the lab, has imaged from these ventral PFNs in the protocerebral bridge. And we think we see sort of these two bumps that correspond to the heading signal there as well. So we think that these what these PFNs are doing, these ventral PFNs, is encoding wind direction in the same format that Larry talked about earlier today for optic flow, where the direction of the wind is not being represented as a compass, by sort of which neurons are activated, but rather the activity of these two populations of neurons sort of projects the wind direction onto a set of orthogonal basis vectors that are really representing these two directions at plus and minus 45 degrees to the fly's heading. And we also think that sort of analogous to what has been shown for optic flow, these neurons are getting their wind direction input through this area called the LAL. So we recorded, we imaged from a set of neurons called LNA neurons that provide input from the LAL to these PFNs. And they also have very strong wind direction tuning that's shown in this yellow curve here. They're not modulated by odor particularly, which is shown in this dark bar here, just like the ventral PFNs. And so we think you essentially have this long pathway, this 5 synapse pathway from the periphery all the way to these columnar inputs to the fan-shaped body that's encoding a pure direction signal. Now, one of the things that was sort of surprising to us was why you maintain this direction signal independent of odor so deep into the brain. And one of our hypotheses about this gets back to this idea that wind direction is kind of an abstract quantity out in the world that you need to derive, and that the way that you derive it is going to depend on whether you're walking or you're flying. So as I mentioned earlier, a walking fly can derive in a very straightforward way the wind direction from movements of its antenna, but if a fly is in flight, it can no longer do that, and it has to sort of compute the wind direction from a comparison between the optic flow that it observes and its sort of expected direction of motion. And this is where we think that this sort of unified basis vector format that we observe both for airflow sensitive PFNs and that Gabby Mayman and others have observed for optic flow PFNs might be very useful, because by putting both of these signals into a format of vectors, as Larry discussed, this could enable the fly to perform these sort of vector computations that it would need to do to derive what the wind direction is when it's in flight. And this is sort of a hypothesis that we're working on testing now. But again, our sort of working hypothesis is that these FB columnar inputs are encoding flow signals or direction signals in a format that allows for a very abstract computation of wind direction that might be able to work both for a walking fly and a flying fly. And that this might allow flies to sort of generalize knowledge that they learn, for example, when they're walking, to a context of flight. So let me just summarize this, this middle part of my talk. I talked about how wind direction is computed in the fly brain. I showed you that a walking fly computes wind direction by comparing displacements of the two antennae, and that there are central neurons that compute this difference by integrating signals coming from the ipsilateral and the contralateral side of the brain. I showed you that columnar inputs to the fly navigation center encode wind direction independent of odor. And I showed you that both airflow and optic flow are represented in a similar basis vector format in these columnar inputs to the fan-shaped body. And we hypothesized that this unified format could allow for an abstract computation of wind direction that would function both in walking and in flight. Are there any questions about this middle part of the talk?
Oh, I guess not.
Okay. Feel free to post questions if you have some. So then in the last part of my talk, I want to talk about how does odor impinge on this circuit, and where does odor actually impinge to control wind orientation. And this is the work of two graduate students in the lab, Andrew Matheson, a student who just finished, and Aaron Lance, sort of a newer graduate student in the lab. So in the first part of my talk, I talked about this sort of odor-guided anemotaxis as one of the fundamental components of olfactory navigation. And what this means is that odor must be able to sort of flexibly and on short timescales change the way that a fly uses the wind direction and turns it into a navigation signal. So in the absence of odor, I showed you that flies like to go downwind. So in the absence of odor, wind is somewhat aversive, whereas in the presence of odor, wind becomes temporarily attractive, okay? As discussed, can the resolution of wind detection be dependent on the velocity of wind? That's a really great question, and we haven't examined it in great detail, though Christina May, a postdoc in the lab, is trying to look at this now. I would say that because the computation depends on a difference between these two displacements, there's some sort of normalization for intensity that happens already in that computation. But sort of, again, quantitatively saying, what's the acuity of the system, what's the resolution with which the fly can resolve wind direction, and how does that depend on the wind speed? We haven't looked at that yet, and that's a great question. Okay, so Andrew set out to identify olfactory circuits that might influence orientation to wind. And to do that, he took advantage of this high throughput wind orientation, wind navigation paradigm that a friend had built, but he adapted it so that instead of presenting odor to the fly, we could activate neurons using optogenetics. And so to test whether this was going to work, Andrew activated olfactory receptor neurons at the periphery and asked whether optogenetic activation of olfactory receptor neurons could recapitulate the behavior that we see when we present odor. And he found that it does. So here he's showing data from the same flies. So in the same flies, if we present apple cider vinegar or we optogenetically activate a large number of olfactory receptor neurons, we get very similar behavior. So in the presence of odor or during activation, the flies accelerate, they turn and run upwind. And when the odor stops or the optogenetic activate lights shut off, the fly will start a search behavior. You can see if you look closely at this, there's some subtle differences between these two. Actually, we see that the odor provides a stronger sort of upwind drive, whereas optogenetic activation generates sort of a stronger offset search, suggesting that these two behaviors are sort of differentially regulated at the periphery. But broadly, we can get sort of qualitatively similar behaviors using both optogenetics and odor. And that's just quantified here. So both apple cider vinegar and optogenetic activation drives a strong upwind movement phenotype, whereas if we just use the parental lines, we see no upwind or movement, or if we drive the crimson using an empty GAL4. We can also show that olfactory receptor neurons are necessary for this behavior. So if we silence all olfactory receptor neurons with tetanus toxin, the flies no longer go upwind in response to odor, and they no longer show this search behavior. And that's quantified here. Okay, so this just sort of establishes that this paradigm works and that you can use optogenetic activation to identify olfactory neurons that might be in the pathway to drive this upwind orientation. So Andrew next turned his attention to these higher order olfactory areas that I discussed on Friday, the mushroom body, which is involved in olfactory learning, and the lateral horn, which is thought to be involved in innate olfactory behavior. And several output neurons of these structures had been identified by other labs that were shown to drive attraction. That is, when those neurons were activated, the flies preferred to be in the light. And so Andrew wondered whether any of these neurons could also drive upwind orientation. So he activated all of the neurons that had been shown to drive attraction phenotypes in these two areas. And what he observed was a subset of those neurons very strongly drove the same sort of suite of behaviors that he saw with peripheral activation of olfactory receptor neurons. That is an upwind run during activation and local search at odor offset. And that was true both with one of the main attractive outputs of the lateral horn, this group of neurons called LH81B2 that drives a strong upwind run during activation and offset search. And also with a cluster of vertical lobe M-bonds, which I talked about on Friday, and these promote attraction. And when these are activated, the fly also runs upwind and searches at a light offset. Okay, so we can drive these behaviors with activation of higher order olfactory neurons, but are these neurons themselves encoding a direction signal, or are they simply encoding the presence of odor that gets integrated with a direction signal downstream? So to look at that, Andrew combined Marie's wind manifold with calcium imaging and imaged odor responses to odor delivered from different directions to the fly in these lateral horn and mushroom body output neurons. And what he observed is that these neurons all responded to odor, but they didn't care about which direction the odor was presented from. And that was true for these two that drove the strongest phenotypes, which I'm showing here. And it's actually true for all the neurons he showed that drove any kind of upwind phenotype. So there are 4 lines he found that drove some upwind phenotype. And here for comparison, he's plotting direction tuning in this LNA input to the columnar system that I talked about in the middle part of my talk. So it seems that these higher order olfactory neurons can drive upwind orientation, but are not encoding a direction signal themselves. They're encoding some kind of pure odor signal. So where might odor interaction signals be integrated? And of course, we hypothesized that this might happen in the fan-shaped body, since we had seen that there were inputs to the columnar inputs to the fan-shaped body that strongly encode wind direction. And there were a couple of connectomics papers that came out in the last year showing that there were a different set of inputs to the fan-shaped body called tangential inputs. These come in from the dorsal brain and go all the way across the fan of the fan-shaped body. And a number of these seem to be anatomically downstream of both the mushroom body and the lateral horn. So to confirm this, Andrew used a technique called transtango, which lets you genetically label the downstream partners of a group of neurons that you're interested in. And when he did this on both of his strongly upwind promoting lateral horn and mushroom body lines, he saw a signal consistent with sort of tangential inputs to the fan-shaped body here and here. So he next set out to identify olfactory inputs to the fan-shaped body. And he and Aaron did this in 2 different ways. One was using a genetic strategy. So they performed a large optogenetic screen of inputs to the fan-shaped body. And what he had found was that there were multiple tangential inputs, both targeting the dorsal fan-shaped body as well as the ventral fan-shaped body, that could drive upwind movement. However, none of the columnar inputs, the PFNs to the fan-shaped body, drove these changes in wind orientation. And these are some controls just showing that if he drives behavior using an empty driver, nothing happens. The fly's behavior is not altered. And in parallel to this, we took advantage of these new connectomic resources, which came out during this study, to identify neurons that might be downstream of both the mushroom body and lateral horn neurons we had found that produce wind navigation phenotypes. And this neuron called FB5AB really stood out. It's the one fan-shaped body input neuron that gets input from all of the M-bonds that drive wind navigation phenotypes. And it also gets input from this lateral horn group that's 2-stage input. As well, Aaron looked at all the fan-shaped body inputs. You can see that there's many fan-shaped body inputs, and looked at their predicted odor tuning by measuring, because we know the glomeruli that encode this attractive odor, apple cider vinegar, that we were using. He could make predictions about which fan-shaped body inputs get the most input from those glomeruli, and therefore would be predicted to strongly respond to apple cider vinegar. And again, FB5AB stood out as having the largest number of connections from glomeruli that would sense apple cider vinegar. So Andrew then asked whether these neurons can drive upwind movement and respond to odor. And so what he observed is that there are multiple fan-shaped body tangential inputs. I'm illustrating 3 of them here, but that we actually found 4 different lines. You can see that they all share this sort of pattern where they innervate a whole row of the fan-shaped body. And all of these, when activated, drive upwind movement. And they also all respond to our attractive odor, apple cider vinegar. And interestingly, just like the lateral horn and mushroom body neurons that I showed you, these neurons don't seem to show very strong directional tuning. There might be a little bit of frontal tuning, but overall, the responses to odor are similar regardless of which direction the odor is presented from. So the picture that this is presenting to us is that the fan-shaped body receives 2 different sets of anatomical inputs that are very distinct in their function. It receives columnar inputs that encode wind direction independent of odor, and it receives these horizontal or tangential inputs that can drive wind orientation behaviors and encode odor, but don't care about the wind direction themselves. And so what this sort of strongly suggests is that wind and signals must be integrated within the fan-shaped body itself. And so that was what we set out to find. Our wind and odor signals actually integrated within the fan-shaped body. And to do this, we also took advantage of connectomics. So because we knew neurons that encoded a pure odor signal and neurons that encoded a pure wind direction signal, we could look for local neurons within the fan-shaped body that get input from both of those populations. And again, there was sort of one population that jumped out as receiving the most input from both of those 2 sources. And these are a group of local neurons called H-delta C neurons. And like the H-delta B neurons that Larry talked about in his talk, these get input in one column of the fan-shaped body and project half across the fan-shaped body, which corresponds, if we think about these as encoding vectors, to a 180-degree rotation of whatever they're representing, okay? And as I said, these get input both at their dendrites from the wind-sensitive PFNs, the ventral PFNs, from both populations. And they also seem to get strong input from this neuron FB5AB, which strongly encodes odor and can drive upwind orientation. And interestingly, the FB5AB synapses onto H-delta C primarily target the output axons of these neurons. And so we think they may be poised to gate the output of these neurons. So do these neurons functionally integrate wind and odor? To address this, Andrew imaged from H-delta C neurons. You can see that they have this columnar structure. So there's about 20 of these, and they tile the columns of the fan. And so if he images from a single column, what he observed now were responses to odor that only responded to a particular direction. So for example, here's one that preferred odor delivered from the front, and here's one that preferred odor delivered from 45 degrees ipsilateral. So overall, each fly seemed to show a different direction tuning, but this is sort of the first place in the system, deep in the center of the brain, where we observe the integration of these 2 signals, odor and wind. Can these H-delta C neurons actually drive changes in orientation? So to look at that, Andrew developed split Gal4 lines that selectively label these H-delta C neurons. And when he activates these neurons, it drives turning behavior, which is consistent with the idea that these neurons, when activated, can cause the fly to reorient. So our sort of working model here is that odor and wind pathways meet in these H-delta C neurons of the fan-shaped body, although they may also be integrated in other places. But how is this circuit actually able to alter behavioral orientation to wind? So here, again, we turn to connectomics, and we had sort of developed a computational model of what we think may be going on, and we're working now to test this model. So coming back to this diagram, if we think about the flies that go downwind in the absence of odor and upwind in the presence of odor, what we can think of this as being is that odor is altering the mapping between wind direction input and motor output. And we can see this quantitatively if we just look at our behavioral data. So if we look at which direction do flies turn at the moment that either odor hits them or that they lose the odor, as a function of which way they are oriented with respect to the wind, we can get these sort of orientation tuning curves. So at the moment that odor hits, if wind is arriving from the right, the flies tend to turn to the right. And if odor is arriving from the left, they tend to turn to the left. And overall, that will stabilize an orientation that is upwind. In contrast, if we look at the moment when flies lose the odor and we look at which way they turn, now if wind arrives from the right, they tend to turn to the left. And if wind arrives from the left, they tend to turn to the right. And so this will tend to stabilize downward orientation. And so we hypothesized that what might be happening in this circuit is that there might be competing pathways from wind direction input to steering motor output, and that odor might allow you to switch which of these pathways is dominant. So we next looked into the connectome to see if there was evidence supporting this model.
There's a question.
Oh, yeah. Could the difference in orientation tuning between flies be due to the involvement of a third reinforcing signal in the mushroom body? Yeah, so there's evidence from, for example, Vanessa Ruda's lab that modulating dopamine signaling in the mushroom body can alter sort of the strength of orientation to wind. And I think that's consistent with our model in which the mushroom body is basically providing an odor value or an odor drive signal. And so if dopamine is gating input onto those mushroom body output neurons, as you turn that up and down, you're basically going to strengthen or weaken the wind orientation signal. I think differences across flies could very much be a result of this. But the difference between the moment within a single fly when it orients upwind versus not, we don't think that switch is happening at the level of the mushroom body because the mushroom body itself doesn't seem to be encoding a wind direction signal. Does that make sense? Great. So how do we think about what the fan-shaped body is doing here? So Larry introduced this idea that we think the fan-shaped body is representing signals as vectors, where the direction of the vector is represented by the location of a bump of activity, and the magnitude of a vector is represented by the sort of the intensity of that activity. Okay. And I showed you in the middle part of my talk that these PFN inputs to the fan-shaped body encode wind direction as the activity of two basis vectors. Okay, and so those neurons will sum onto their outputs in the fan-shaped body and produce a single bump of activity whose location and intensity represent the direction of the wind. Okay, and when we looked at the connectome, there seems to be a direct pathway from these wind direction inputs onto neurons that have been suggested to be involved in steering, called PFL3s. And PFL3s are poised to basically look at a bump of activity in the fan-shaped body and compare it to a left or right turn and say, would a turn to the right, or a turn to the left bring me closer to my goal? And if we look at this bump of activity produced by the PFNs, this should promote downwind orientation. So if we project this bump of activity in the PFNs onto right and left PFL3 neurons, then if wind comes from the left, the projection onto the right PFL3 is larger. And so wind coming from the left should produce a right turn, which should produce downwind orientation. And so we can sort of propagate this signal through our model and get a predicted tuning curve for this direct PFN to PFL3 pathway, which has a downwind orientation, much like the downwind tuning that we observe in flies behaviorally during the absence of odor. So how does odor enter the system? I showed you that these H delta C neurons receive odor input at their output tufts, and moreover, they turn on during odor, right? So they're activated by odor. And these H delta C neurons project halfway across the fan-shaped body, which would correspond to 180-degree rotation of any vector that they represent. Now, the H delta C neurons also receive input from wind-sensitive PFNs, so they should be encoding a wind direction signal. And then they project halfway across the fan-shaped body where they project again onto these steering-related PFL3 neurons, but through an intermediary called FC neurons. And so the prediction here is that what the H delta C neurons should do is they should take this wind direction bump represented in PFNs and rotate it by 180 degrees. And now if we compare this bump to the left and right PFL3 neurons, you can see that this bump is closer to the left PFL3. And so now wind from the left is going to promote left turns. And again, if we propagate this through our model, this generates an upwind tuning curve consistent with what we observe behaviorally in the flies in the presence of odor. So our hypothesis is that there are these direct and indirect pathways within the fan-shaped body that allow for rapid context-dependent switches in wind orientation. The direct pathway from wind direction input to steering motor output promotes downwind orientation, whereas the indirect pathway rotates this bump by 180 degrees to promote upwind orientation. And we're working on testing this model now. So just to summarize this last part of my talk, I showed you that in addition to this wind direction pathway, which encodes a pure wind direction signal, there's an olfactory pathway in the fly brain that drives upwind orientation, but encodes a non-directional odor signal. I showed you that the fan-shaped body receives both pure odor signals through tangential inputs, as well as pure wind direction signals through its columnar inputs. And I showed you that H delta C neurons within the fan-shaped body receive input from both pathways and can drive locomotor changes. And I put forward a hypothesis that these competing direct and indirect pathways within the fan-shaped body could promote downwind and upwind orientation respectively. And we hypothesized that odor gates the activity of the indirect pathway on to promote upwind orientation. Okay, so just to summarize some of the take homes from my talk as a whole, as well as some future directions that we're looking at now. I've shown you that olfactory navigation is a highly tractable ethological behavior. It's a really rich behavior where there's a complex physical stimulus that flies have to translate into a series of motor actions in order to reach a goal. I've shown you that we've identified sensory pathways that compute, encode, and integrate the two sensory stimuli that we believe are required for olfactory navigation, that is the identity of the odor, as well as the direction of the wind. And we're currently testing the hypothesis that these inputs to the fan-shaped body encode rather abstract internal variables, such as the odor value or the wind direction, in a manner that's somewhat removed from the actual peripheral sensations that the fly receives, and that would allow for certain generalization. For example, if these neurons encode an abstract wind direction signal that works in both walking and flight, then if a fly learns using its mushroom body that a new odor should be associated with food, it can immediately generalize that to a condition where it's flying without having to learn a new mapping between a different set of sensory inputs. Another thing we're working on now is trying to identify the motor circuits that translate these navigational commands that we think are coming out of the fan-shaped body into locomotor actions, partly by taking advantage of this set of genetic tools for labeling descending motor neurons. And finally, a direction that we're sort of thinking about, I think Antonio introduced the idea, I hope, that there are many different models in the literature of how, so algorithmically, animals might use odor information to navigate. And interestingly, they range from sort of very simple models that are essentially a series of chained sensory reflexes, like this odor-gated anemataxis, to computationally very intensive models like infotaxis that require sort of a large amount of computational processing power. And I would say it's been challenging using behavior alone to identify which algorithms the flies are using and to tease apart sort of how computationally complex is the thing that they're doing. And one thing that we hope in the future is that these fly circuits that we're starting to work out can provide sort of a circuit and biophysical basis to ask these questions more precisely. So the pathway that I focused on today is actually a very long pathway through the brain, and this was somewhat of a surprise to us. So I've shown you that there are pathways that are multiple synapses long that encode a pure odor signal as well as a pure wind signal, and that we finally observe integration of these two signals in local neurons of the fan-shaped body, which then can provide motor output. But some of the work that we're doing, especially looking at motor neurons, as well as data from the connectomes, suggests that there's probably parallel pathways in the system as well. That is, there are shortcuts from odor encoding areas to wind encoding areas, and multiple pathways, particularly from these wind encoding areas to motor control. And what this really suggests to us is that this is a circuit that doesn't have one fixed architecture with a certain number of layers, but instead has sort of a parallel architecture with both very short sensory motor loops and very long sensory motor loops operating in parallel. And one of the sort of questions that we'd like to address in the future is whether we can use this architecture in the fly brain to understand how both simple and complex algorithms might be working in parallel in the fly in order to optimally navigate in a variety of odor environments. So I'm going to stop there. I'd like to thank the people who did this work. This was largely the work of sort of the first generation of Nagel Lab alumni, postdocs Efren Alvarez Salvado and Marie Suver, and graduate students Tim Currier and Andrew Matheson, as well as Andrew Lance, a current graduate student in the lab. We've been really fortunate to collaborate with a number of really excellent labs, including John Grimaldi's lab, who are fluid dynamicists at the University of Colorado Boulder, who made the plume measurements. Jonathan Victor, who's a computational neuroscientist as well as a visual neuroscientist at Weill Cornell. My husband, David Shopik, with whom we work closely in his lab. And Mubarak Sayed, who's a developmental biologist interested in fan-shaped body development. We've also been really lucky to take advantage of all of these wonderful fly tools and resources, which I talked about in my previous talk. So the many fly repositories, as well as connectomic resources, as well as our funding sources. So I'll stop there and I can take any questions.
Thanks, Kathy. Any questions? Okay, let me start with one. So in flying flies, when they experience wind frontally, they pitch down. So when they pitch down, they actually sort of changing the optic flow on their retina, and maybe also the antennal angles. Do you think there's any sort of changes in gain that go on that account for this kind of pitching forward?
That's a really interesting question. So to my knowledge, people really haven't thought about how these central complex circuits are working in the third dimension of pitch. Yeah. And to be honest, we're sort of just starting to think about this. So kind of our working hypothesis is that there are local neurons in the fan-shaped body that are getting input from both these optic flow and airflow sensitive PFNs, and can combine it in sort of a clever way to potentially encode something that would remain stable. For example, so we've been talking to both John Grimaldi as well as Floris van Breugel about like what do flies actually experience when they're hit by a gust of wind, and maybe you know more about this. So it'd be great to talk about this, but it sort of depends. I mean, so you can sort of have sort of a physics model of pretend the fly is a particle and it's just moving forward. Yeah. And in that case, when it's hit by wind, it'll initially experiencing a transient mechanosensory stimulus, and then a more long-lasting optic flow stimulus as the fly gets moved by the wind. And so under those conditions, we think that some of these local fan-shaped body neurons may be able to generate a signal that's relatively stable. Now, of course, the fly is actually controlling its response. And so it sort of depends on what you think the fly's sort of control theory response is. And so we've also been talking to Flores, and he has sort of a variety of different models, which all produce different things. My prediction would be that some of these fan-shaped body neurons would also encode changes in pitch, but we don't have any data on that yet. So I don't know if I can speak to that. Do you have thoughts about exactly what a fly experiences when it's hit by wind while it's in flight?
Something similar to what you just said, which is a transient response, a transient gust, and then something more stable. That's pretty much what I think is going on. And then, to some extent, they get swept by the wind, or they respond to it, and so then it becomes complicated. But yeah, I mean, the transient part is something that I'm sure happens.
Yeah, and I mean, if the wind is self fluctuating, then there'll be a transient presumably for every fluctuation, right?
Yeah.
We have seen that when we let the flies orient to an airflow stimulus in flight, they will orient to it. So they can do that. I mean, they do something that doesn't make a lot of sense, which they tend to go downward, which is also what they do if they're given free control of optic flow during flight. So the tethered flight situation is a little hard to interpret at the moment. But yeah, we're hoping that this will be sort of an interesting area to think about, and it always seems sort of intractable to me to think about how they might be doing this computation, but this idea that the fan-shaped body represents and encodes things as vectors seems to open you up to doing the right computations. So it's essentially
one of the things that comes out of your talk, which I thought was very interesting, is that for the longest time we've been trying to speculate about why insects keep their antennae at the angles that they keep. And we found, at least in moths, also in flies, that when they experience frontal wind, they will actually change their antennal angles. And this happens also in bees, for instance. So they're actually changing the antennal angle in response to the airflow. And so it's not clear, I mean, given what you've just told us, whether this could explain what they're trying to do by changing those antennal angles. I mean, the response is very, very, very clear.
Yeah. So we have a new study which I didn't talk about, but we're just writing up now. So Marie Suver got interested in how they move their antennae.
Yeah.
And she and an undergrad in the lab were able to find motor neurons that move the antennae.
Yeah.
And so we can activate those motor neurons and then look at how that changes wind encoding. And it looks like they can basically change the gain of wind encoding by moving their this way or this way, which is what you predict. And we observed this a similar thing, both that they change their position and that they wiggle them a lot more when there's sort of more wind. And our hypothesis is that in fact it's almost like a foveation mechanism, right? That if you move them forward and up, you get more acuity for like small wind currents directly in front of you.
Right.
Whereas if you move them back, you could have sort of broader sensing of wind from many angles. Does that sort of fit with your observation?
We don't know. I mean, we speculated something like that, meaning that what they're trying to do really is optimize their ability to sense an airflow depending on how fast they're flying, optic flow, etc. We know that optic flow also causes them to change their antennal angles, and it's still in the opposite direction of airflow. So if front to back optic flow will cause the antennae to go back, and front to back airflow will cause them to go front. So the two counteracting things going on when a fly is flying forward. We really don't know beyond that what might be going on. We always felt that this somehow, between these two counteracting responses, the antenna stays, at a given angle, and that's what the insect is using then to kind of make sense of, I don't know, make sense of its optic flow vis-a-vis its airflow that it's sensing. But
do you see a difference in the gain of those responses for flies which have these short stubby little antennae versus moths that have much longer antenna, or are they doing sort of a similar thing?
yeah, I haven't looked at flies at all, only moths, bees, but both of them show the same responses, both moths and bees. Yeah.
Yeah, interesting. Do you have an idea what they're doing with these sort of quick movements? Because we see a lot of those, and we also see that they increase these in the presence of wind, and that we think they're active because if we silence the motor neurons, they're decreased.
Yeah.
But I don't know
I mean,
exactly.
there's little flicking movements, right?
What's that?
Little flicking movements, right?
That's right.
Yeah, yeah. And the standard idea is that they're flicking off the boundary layer to get new odor, but I don't know.
Yeah, so what we see, which was interesting, is that they didn't increase these in response to odor at all.
Right.
But they definitely increased them with wind.
Yeah.
So we're trying to make sense of that.
Interesting. Let's see if there's any other questions. Yeah, there is a question.
Do you also have studies on how their brains somehow keep a memory of the information they collected when they track sense and odor patch? Yeah, so I think that's a really interesting question. We haven't looked at this yet, but this again gets to the question of why is this circuit going through the fan-shaped body at all? And I think that it's still a really open question in fly neuroscience, which behaviors require the central complex and which do not. And so I think it's a reasonable hypothesis that part of what these circuits are doing would be either developing some internal estimate of where the fly thinks the source is based on this sort of stochastic input that's coming in from the plume plus their own path integration. Or it might help keep a wind direction memory or something like that. And we haven't looked at that directly yet, but I think we have the tools now to ask whether that is happening. And I think that's something we'd be very interested in asking in the future. That's a really good question.
Yes, there are no other questions. And it's 8:30 here. So thank you, Kathy. That was an absolutely wonderful talk. Both of
Thanks.
them. So yeah.
Yeah, if anybody has any other questions, feel free to ask me in Slack, and I'm happy to chat further with anybody.
Yep.
And thanks again for inviting me.
Oh, it was wonderful. Thank you, Kathy.
All right.
Bye.
Thank you. Bye.